Okay, folks, so we are going to just have a look at what happens in the nephron. So before we do that, we just need to recap what it means for diffusion, osmosis, and active transport. So we're continuing on in your notes book. So both diffusion and osmosis are examples of what's called passive transport. So hopefully you are now saying, Miss, that is when no energy is needed and it is going from high to low concentrations, whether that is water or sugar or salts. So diffusion is the top diagram here, that if you spray some deodorant, the smell will spread out. And our higher level definition is movement of molecules from an area of high concentration to an area of low concentration, and it is along the concentration gradient. Whereas osmosis specifically refers to water, and in our definition it's still from a region of high to low concentration, but we're talking about water and it needs to move through the semi-permeable membrane. So when you come back, we'll do our osmosis experiment. This here is our visking tubing. It's the semi-permeable membrane that we're going to use. It's artificial, and if it was a cell, it would be the cell membrane, would be the semi-permeable membrane. So there's two examples of what's called passive transport. So they just happen because the concentration conditions are right. And if it's not passive transport, it's called active transport. And that's when energy is needed. And you're now moving from low to high concentrations and you're going against the concentration gradient. So that will happen in our nephron. Now we will see active transport being used. So our nephron is a filter. It filters our blood. And when we drew our diagram the last day, we drew a cup, a squiggle, a loop, another squiggle, and a candy cane. And we drew a blood supply. So what we're going to talk about is what happens here. It's called in terms of filtering the blood. And then how does all of the stuff that gets filtered go back into your blood? So it's called reabsorption. How does that happen? So you need to know your parts of your blood. So we've said urine is made in the nephron, and the three main processes that we're going to look at today is filtration, reabsorption, and a thing called secretion. Now you have this table, and we're looking at the part of the nephron that we're looking at is the glomerulus. So that's all of these little capillaries portions that are here. And what happens is the blood is coming in here. When it gets into the glomerulus, because it's one cell thick, everything gets squeezed out of it that can possibly get squeezed out, and it goes into the nephron at the Bowman's capsule. And the reason it gets squeezed out is that you'll notice down here this afferent arteriole is really, really thick compared to this efferent arteriole here, which is much skinnier. So therefore the blood that's coming under in this side is under really high pressure, and that's why stuff gets squeezed out of your blood. So in our glomerulus, you have to know is what is filtered from the blood, so what leaves, what stays in the blood, what is not filtered, and how is this glomerulus, this whole load of capillaries, adapted to be really, really good at its job. You also need to know what is the name now of the stuff that once it gets into this Bowman's capsule here, what is the name now of the solution that's in there. So what gets filtered from our blood? Loads and loads of useful stuff. So the glucose, amino acids, vitamins are the useful stuff. You will have some hormones. So we've said that if you need to pregnancy test, you can wee on a stick now and it'll detect some hormone for you. And these are our excretion products. The urea, the salt, and the water are the products of excretion. The things that can't leave the glomerulus, that can't get into the Bowman's capsule here, are things like your red blood cells and your white blood cells and your platelets, antibodies, and some hormones. And the reason for this is because these are very big. They just can't get through the one cell thick. So they literally don't fit through the filter, so they stay in it. And the glomerulus adaptations, what makes the glomerulus really good at filtering, we've said, is the pressure is greater than normal. And that's because this efferent arteriole is narrower than the afferent. So This part here is much narrower than this, and that causes your high pressure. We also have the advantage because we have loads of blood cells in here. I'm colouring in red because I can't change my colour pen, and that provides a really large surface area. These capillary walls are more porous, so they are very leaky, they'll allow things in, and the Bowman's capsule is 1 cell thick, so that also helps. So our capillary is 1 cell thick and our Bowman's capsule is 1 cell thick, and those two things together mean that all of the things that can leave the blood will leave the blood and be squeezed into your Bowman's capsule. So once the stuff is in our Bowman's capsule down here, it is called glomerular filtrate because it's come from the glomerulus and it's been filtered. So that's the stuff that contains all of the things, that with the good stuff and the bad stuff. And a question that the SEC can ask is, how does the glomerular filtrate different from blood? And it's the fact that it's all the big things. So there'll be no red blood cells, no white blood cells, no platelets, and no big antibodies. And again, that's because these guys are too big to be filtered. Okay, so we've done our filtering now. We now have to do reabsorption, and when we do our reabsorption, getting stuff out of the nephron into our blood, we're going to use one of these 3 methods in which to do it. Now, this is in your notes book, so you have to be able to tell the SEC where and how much water is absorbed back into the blood and what's the process it uses. So I use this little silly sentence, OMANSA, and the O is obviously osmosis, and the M stands for most, the A is a little, the N is for none, the S for some, and the A is a little. And the letters follow the sequence of our nephron. You also have to tell them how much of the good stuff is reabsorbed. So this is not only the salt, it's the glucose, it's the amino acids, and all of those things. And my silly sentence for this is, my nan serves sizzling noodles, and I'm interested in the first letters. So again, the M is for most, the N is for none, S for some, S for some, N for none. And again, you have to be able to match the letter to the part of the nephron. In reality, if you look at the questions, everything that gets reabsorbed happens in your proximal convoluted tubule. But in case they ask it differently, OMANSA and my nan serves sizzling noodles will help you figure it out. So we've done our filtration up at this portion, so that's now gone, and we're now interested in the squiggle, the loop, the next squiggle, and the candy cane for reabsorption. So what do you need to know? Vast majority of stuff that got squeezed out of our blood gets reabsorbed. So it's going to use everything else. So this is the squiggle, these guys are the loop, so this is the squiggle. These guys are the loop, this is the other squiggle, and this is the candy cane. So when we're going to talk about them, we're going to talk about all of the different parts. So you have this table, and you will know that there'll be nothing here, nothing here, and nothing here, and we're going to use, as we said, OMANSA and how my nan serves sizzling noodles to do this. So looking at our water, for using OMANSA, most of my water is going to be in my proximal convoluted tubule. I will have a little in my descending loop, and I will have none in my ascending loop. I will have a little or some in my distal convoluted tubule, and I will have a little water reabsorbed in my collecting duct. So if we were to draw our bits here, squiggle, I did none that wrong, haven't I? I think it'll erase for me now. And candy cane, this is the most, this is the A for a little, there is none here, there is some at the squiggle here, and there is a little for our collecting duct. So M-A-N-S-A. And again, because it's water, there's only one way it can be, and again the question will look for the process, it has to be osmosis. So that is the only way water gets moved around in cells. So So then we look at all of the good stuff that's in the blood that we want to get back out. So it's the Mynan serve sizzling noodles. So again, most of the stuff is going to happen in your proximal convoluted tubule. There is none in your descending loop. There is some in your ascending loop, some in your distal tubule, and none in your collecting duct. And you will find that most of the process by which it happens is diffusion, because there'll be high concentration in the nephron and a low concentration in the blood. But active transport will kick in at the end of your proximal convoluted tubule, and again at the very, very top of your ascending loop. So the ascending loop is when the loop starts to turn up toward your distal convoluted tubule. So again, if we were to do cup, squiggle, candy cane, squiggle, collecting duct, and we're now looking at the salt, this is the most in the squiggle. There is none here. There is Mynan serves, some here. Sizzling noodles, we're going to get some here, and none in the collecting duct. So Mynan serves sizzling noodles. And as I say, the question that they usually ask is, the answer is nearly always proximal convoluted tubule. Now there's little asterisks here, and you have to know how these structures are adapted to their functions. So in terms of how they're adapted, our proximal tubule is 1 cell thick. It's very long. We draw it as a squiggle, but it's really, really squiggly. So there's loads of time for things to get reabsorbed, and there's lots of mitochondria to help with active transport if that's what's needed at the very end of it. The descending loop doesn't have any adaptions, but the ascending loop, you will get loss of salt into the medulla, will allow osmosis to happen, and that is a good thing. In your distal convoluted tubule, osmoregulation may happen here, so you're moving salt in and out of your nephron to make sure that osmoregulation is maintained. And in your collecting duct is adapted to remove water if it needs to, because its walls are permeable to water. Now, we've seen our kidney before, and just to have the last little reminder, the liquid leaving in the collecting duct, so this is our nephron, squiggle, candy cane, squiggle, collecting duct. So all the collecting ducts end up in here in your pelvis, and then the urine will flow down your ureter into your bladder. So once it gets into the pelvis, you now have urine. Now, in terms of secretion is just when actually substances are going from the nephron or from the blood into the nephron, and they do it at the distal convoluted tubule, and that is going to help with osmoregulation. So this is why they're doing it. So there's trying to get the salt balance right and water balance in your blood right, and to do that it pushes things into your blood for osmoregulation. And another question the SEC can ask is not only how does the glomerular filtrate differ from blood, but how does it differ from urine? So essentially the glomerular filtrate will contain more water, because water is going to be reabsorbed before it gets to and whatever's left is going to be going to the urine, and your glomerular filtrate compared to urine contains more useful stuff. So the glucose, the amino acids, and the vitamins. If you have glucose and amino acids in your urine, there's something wrong, and you will require medical help. So if you have glucose in your urine, it's usually a sign of diabetes, which is not good. And amino acids, you can sometimes get these in your urine when you're pregnant, and again, that is not good, so it's a sign of something else. I think that pretty much leaves us now to the end of this. I am on Zoom if you have any questions. This is tough stuff, but if you can learn your silly sentences and you can match them to the part of the nephron, then you can answer any question. The only thing you have to add to it then is the adaptations of your glomerulus and your Bowman's capsule, and your adaptations of the different parts of your nephron to the jobs that